Hello dear students, Mendelian disorders and chromosomal disorders. Imagine if a hemophilic person has to undergo surgery. Here you can see the normal blood vessels. Head may be smaller than the normal, hence we use the word called microcephaly. This Turner syndrome is common in whom? It occurs only in females. Hello dear students, welcome back to session 9 of this very important chapter called Principles of Inheritance and Variation. And this is the last session of this very important chapter called Principles of Inheritance and Variation. Hope you remember in the last session, that is session 8, we studied about mutation, what is mutation and what are the types of mutation, that is the gene mutation, chromosomal mutation and frame shift mutation, and also we studied about pedigree analysis, right? So in today's session, what are we going to study? Hope you remember the genetic disorders are studied in the human society as human genetics, and genetic disorders have been broadly classified into one called Mendelian disorders and chromosomal disorders. That is, gene disorders have been broadly classified into two types. What are they? What are gene disorders? So that which particularly the gene disorders have been broadly classified into two types called Mendelian disorders and chromosomal disorders, right? Now we'll start with the first one called Mendelian disorder. Mendelian disorder is due to the alteration or mutation in a single gene. So what is Mendelian disorder? Mendelian disorders are the disorders that occur due to alteration or mutation in a single gene. Hence even the Mendelian disorders are also called as monogenic disorders. So Mendelian disorders are also called as monogenic disorders. And the one what we are studying here, you can see the title, hemophilia. Hemophilia comes under Mendelian disorder or monogenic disorder. What are the examples for Mendelian disorders or monogenic disorders? We come across hemophilia, cystic fibrosis, sickle cell anemia, Phenylketonuria, then thalassemia. All these are the examples for Mendelian disorders. So what are Mendelian disorders? Mendelian disorders are the disorders that take place due to alteration or mutation in a single gene. And what are chromosomal disorders? Very, very important. That is what I am going to cover in today's session. Both about the Mendelian disorders as well as the chromosomal disorders. The very word itself will tell you. What do you mean by chromosomal disorders? The disorders that happen due to the change in the chromosomal number. Hope you all know that the chromosomal number is constant for a particular species. Either addition or deletion. That is one extra chromosome or removal of one chromosome from the normal set of chromosome is going to alter the chromosomal number, which in turn is going to cause disorders or anomalies or abnormalities, right? So we are going to study about three examples in chromosomal disorder. Moving on to the first Mendelian disorder, that is about hemophilia. Hope all of you know that hemophilia is a gene disorder caused due to the less number of clotting factor, which results in what? Profuse bleeding. So due to the lack of the clotting factor, there will be continuous oozing of blood. Hence hemophilia is also called as bleeder's disease. It is also called as what? Bleeders. Why it is called bleeders disease? Because there is absence. I hope all of you know that when we have an injury or a cut, blood comes out, oozes out, but immediately it clots. So when the blood comes in contact with the atmospheric oxygen, the blood will clot. But what will happen in case of hemophilic patient or a hemophilic person? So in them the clotting factor is absent and obviously there is continuous oozing of blood. Sometimes it may prove fatal. So it is a gene disorder due to the absence of or a less number of clotting factor, which results in continuous or profuse oozing of blood even from a small injury. Hence the name bleeder's disease. And it takes prolonged time due to the lack of clotting factor. So there is, as I said, there is a continuous oozing of blood. And hemophilia is due to the recessive gene present on X chromosome. It is due to the recessive gene present on the X chromosome. Hence it is called as X-linked inheritance. It is called X-linked inheritance. Why it is called X-linked inheritance? It is a recessive gene. It is a recessive gene present on the hemophilia is due to the recessive gene present on the X chromosome. Hence it is called X-linked inheritance. So hemophilia is heritable. It is heritable. What do you mean by heritable? That it is passed on from one generation to another generation. And one very important thing is hemophilia occurs more in male than in female. Why? Why the occurrence of hemophilia is more in male than in female? Because a single recessive gene present on the X chromosome is sufficient to express this character of hemophilia in males. But in females, females should have two recessive gene on the X chromosome to express the character. So it is very rare in females, right? So hemophilia is also, and this hemophilia was the first time discovered by John Conrad Otto in 1803. So this disease called hemophilia was first time discovered by a scientist by name John Conrad Otto in 1803, and it was first observed in the European family, that is in Queen Victoria's family. And the family pedigree or family analysis indicates that Queen Victoria had a number of descendants who had this hemophilia, and it was transferred from one generation to another generation. So what is hemophilia? As I said, it is a sex-linked recessive disorder in which blood fails to clot due to the non-production of certain blood clotting factors. Due to this, in an affected individual, a simple cut will result in what? As I said, due to this, there is a prolonged oozing of blood, and it may, even from a small wound also, you can see the profuse oozing of blood. And sometimes, as I said, it may lead to death. And the heterozygous female carrier for hemophilic may transit the disease to the sons. Usually the females are the carriers and the targets will be the sons. So as I said, why it is very rare in females, why it is more common in males? Because one single recessive gene is sufficient for the expression of the character, that is the hemophilic character in males. But in females, two recessive genes should occur on the X chromosome for the expression of the character. So the possibility of female becoming hemophilic is extremely rare. And this is because the mother of such a female has to be at least carrier and the father should be a hemophilic. So in that case, both XX will have the recessive gene, and then there are chances of female being hemophilic is quite common, but otherwise it is very, very rare. As I said, it was first discovered in the Queen Victoria's family, and she was a carrier of this disease and produced what number of descendants having this hemophilia. So this is about hemophilia. As I said, hemophilia is a classical example for Mendelian disorders. What are Mendelian disorders? They are the disorders which are caused due to alteration or due to mutation in a single gene. Hence the name, it is also called as what? Monogenic disorders. What are the other classical examples for monogenic disorders other than hemophilia? Sickle cell anemia, cystic fibrosis, then thalassemia, all these are the phenylketonuria, all these are the examples for Mendelian disorders. Hope you have understood about what is hemophilia. Moving on to the next very important, I can see the diagram here. See, this is the normal blood. See the beautiful picture, right? Here you can see normal blood vessels. So usually when there is a rupture of normal blood vessels, the bleeding happens. So bleeding there is oozing of blood. But immediately what will happen? See here, you can see the incomplete formation of the hemofibrin, a clotting factor, right? So here you can see it will not clot. So what will happen? See here, completed fibrin clot. So this will, see here it is hemophilic. So incomplete fibrin clot continuous what? Bleeding. Here there is no continued bleeding because of the process of formation of a clot called fibrin. So that is the reason why, imagine if a hemophilic person has to undergo surgery. Usually in the process of surgery there is the loss of blood. So in case, imagine if a hemophilic person has to undergo surgery, then definitely it is very, very difficult. So but before they undergo surgery, they will be induced with hemophilic so that the clotting factor is induced into their body, so that surgery can take place and the person will be quite safe. So hemophilia is something which is very, very serious disease, we can say, right? So you can see in this beautiful picture the normal blood vessel, when it ruptures, clotting will happen here, but here in this case there is no clotting here. So that is the reason there is profuse oozing or prolonged bleeding. Hence it is also called as what? Bleeder's disease. Moving on to the sickle cell anemia. As I said, again it is an example for Mendelian disorder, and it is an autosomal recessive gene. It is due to autosomal linked recessive trait. And here what will happen is in a normal hemoglobin, the RBC will be biconcave, but in case of sickle cell anemia, the RBC cells will be sickle shaped. So what will happen when the blood flows inside the artery? So it is going to rupture the walls of the arteries and lead to anemia because of that sickle shape sharpness. So it is going to cut the blood vessels, right? So it can be transmitted from where? From parents to offspring. Usually the normal person, the normal hemoglobin will be HbA, whereas in a person who is having sickle cell anemia, the allele is GBS. So S is a defective hemoglobin. So S is a defective hemoglobin. Defective hemoglobin. So when HbA is homozygous, then the person will be a normal person. So he or she will not have what? Sickle cell anemia. If HbS occurs in homozygous condition, then definitely the person will be affected with chronic sickle cell anemia. So HbA, HbS. So when if it is HbA, the chances of the person will be a normal male. So the gene for the normal hemoglobin is HbA, whereas the allele for the defective hemoglobin is HbS. So as I said, when HbA occurs in homozygous condition, the person will be normal. But if HbS, the defective hemoglobin, occurs in a homozygous condition, then the person will have chronic sickle cell anemia with having the moon-shaped RBC cells instead of being biconcave. And also one of the amino acid called glutamic acid is replaced by valine. So one of the amino acid called glutamic acid is replaced by valine in case of sickle cell anemia. As I said, it can be transmitted from parents to offspring when both the partners are carriers for, that is heterozygous. The disease is controlled by a single pair of allele called HbA and HbS. So out of 3 possible genotypes, only homozygous individuals for HbS show the disease phenotype. As I said, if HbS occurs in homozygous condition, then the chances of the person having sickle cell anemia is quite possible. So here heterozygous HbA, HbS individuals appear apparently unaffected. So here you can see one is HbA and one is HbS. They are the carriers of the disease. Suppose if it is both HbA, normal. If it is HbS, affected. If it is HbS, these are normal, these are affected, and this is carriers. They will be the carrier. That is, there are 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. This defect is caused by substitution of what? Just now I told you all. Substitution of an amino acid called valine is replaced by, valine is replacing what? Glutamic acid. Position of the beta globulin chain of the hemoglobin molecule. So the substitution of amino acid in the globulin protein results due to single base substitution at the 6th codon of beta globulin gene from GAG to GUG. So GAG will be converted into GUG. So that is valine replaces what? Glutamic acid. So here glutamic acid should be there. But what will happen? Glutamic acid is replaced by valine. Then the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC. As I said, usually it will be biconcave, but here it will be moon shape like this. So obviously when this sickle shape, so you can see the sickle shape. So when it passes through the blood vessel, obviously it is going to cut or rupture the blood vessel, leading to what? Anemia. So biconcave disc to elongated sickle-like structure. So that is all about sickle cell anemia. This is also very important from examination point of view, which is quite commonly asked in the exam. So here you can see normal, you can see about this sickle cell anemia with this beautiful picture. Here you can see the normal blood vessels. See how they are. So RBC flow freely within the blood vessel. How do they flow? Because they are biconcave. So they are not going to rupture the. So here you can see the normal hemoglobin. It is biconcave. You can see here. It is biconcave. So you can see sticky sickle cells here. See, you can see the shape here. It is moon shape. So what will it do? It is going to block the flow of the blood vessels. Why? Because it is going to rupture also, and it is going to block. That sharp ends will come and it is going to stop the blood flow. Here you can see abnormal hemoglobin from strands that cause what? Sickle shape. Here you can see the sickle shape, right? Moving on to phenylketonuria. Again, it is commonly called as PKU-EDS. You may wonder why they are called as PKU-EDS, because they show abnormal mental behavior, hence they are referred to as PKU-EDS. This is an autosomal recessive trait, and it is due to the abnormal metabolism. There is one amino acid called phenylalanine that is here due to abnormal metabolism, I said, because due to the absence of an enzyme called phenylalanine hydroxylase. So the affected individuals lack an enzyme called phenylalanine hydroxylase that catalyzes the conversion of an amino acid called phenylalanine into a substance that is found in the food called tyrosine. So due to the lack of an enzyme called phenylalanine hydroxylase, so what will happen? So phenylalanine is not metabolized. So it remains accumulated, or it is converted into derivatives like phenylpyruvic acid. And these derivatives will go and accumulate in the brain cells, leading to mental retardation. Hence they are also referred to as what? PKU-EDS, right? So consequently, phenylalanine is metabolized into phenylpyruvate and other derivatives. So when there is a lacking of this enzyme called phenylalanine hydroxylase, obviously, as I said, the phenylalanine is not metabolized and it is accumulated, or it is converted into its derivatives like phenylpyruvate and other derivatives. And what will happen? All these derivatives will start accumulating in the brain cells, leading to what? Mental retardation. Hence they are also referred to as what? PKU. They are referred to as PKU-EDS. They are referred to as PKU-EDS. And these are also excreted in the urine, as they are not absorbed in the kidneys. Right? So these are also excreted in the urine, as they are not absorbed by the kidneys. So all these are the very, very important features of phenylketonuria. As I said, this is an example of what? Autosomal recessive trait. As I said, it is also an example of what? Mendelian disorders, which is caused due to alteration or mutation in a single gene. Here you can see the unaffected carrier father, then affected mother. So here if you see the analysis part here, these are the affected. These are the carriers, and this is also carrier. This is also carrier, because unaffected carrier father, unaffected carrier mother, both are carriers, but they are unaffected. So what will be the possibility? 3 will be the carriers and 1 will be affected, as 1x from father, 1x from mother comes. So obviously, so it will be affected. As I said, usually the males will be affected. So this is an autosomal recessive trait. The affected individuals lack an enzyme called phenylhydroxylase that catalyzes or conversion of what? Amino acid called phenylalanine. But due to the absence of this enzyme, phenylalanine is not metabolized and it is accumulated or it is converted into its derivatives like phenylpyruvate and and all these phenylpyruvate and other derivatives start accumulating in the brain cells, results in mental retardation. As I said, these are also excreted in the urine as they are not absorbed. As you know, the nephrons of the kidneys absorb, do the process of absorption, but here it does not do, because these are also excreted in the urine as they are not absorbed in the kidneys. Moving on to a very important concept called chromosomal disorders. Chromosomal disorders, as I said, they are due to either addition or deletion of a chromosome from the normal set of chromosomes. Usually all of you know that the nucleus of a cell will have balanced set of chromosomes called 2n. So neither it should be 2n minus 1 nor it should be 2n plus 1. So this change in the chromosomal number will lead to what we call it as chromosomal disorders, where we also use a word called aneuploidy. Aneuploidy is what? Due to unbalanced set of chromosomes. Why it is unbalanced? What is the balanced set of chromosome? 2n is always the balanced set of chromosome, and this balanced set of chromosome is altered. Why it is altered? Either there is deletion or addition of one chromosome to the normal set of chromosome, Hence we use the word called aneuploidy. There's one when there is a balanced set of chromosome, we call it as euploidy. Right, and one very important example for Down syndrome is, it is an example for aneuploidy, right? So before we go to that, we have to understand what are the types of aneuploidy. What are the types of aneuploidy? One is called monosomy, that is 2n minus 1. Trisomy is 2n plus 1. Tetrasomy, it is 2n plus 2. Nullisomy, that is 2n minus 2. So these are the types of aneuploidies. So this addition or deletion may take place to an autosome or to an allosome. As you all know, there are two types of chromosome. One is called autosome or the body chromosomes, which is constant or common in both male and female. But the change occurs in the allosome or the sex chromosomes. In female it is XX and in male it is XY. So this change in the chromosomal number may take place to an autosome or to an allosome. If at all it takes to an autosome, we call it as autosomal aneuploidy. If at all it takes place to an allosome, we call it as allosomal aneuploidy. It might be hyperaneuploidy or hypoaneuploidy. What do you mean by hyper? Plus 1. What do you mean by hypo? Minus 1. So it may be autosomal hyperaneuploidy or allosomal hyperaneuploidy, or autosomal hypoaneuploidy or allosomal hypoaneuploidy. So let us understand about this Down syndrome. So as I said, Down syndrome is a type of aneuploidy which is due to trisomy. What do you mean by trisomy? Addition. And this is an autosomal hyperaneuploidy. Down syndrome is an example for autosomal hyperaneuploidy. That means it is due to 2n plus 1, but that plus 1 is to the 21st autosome. So instead of 44 plus, and this Down syndrome is common for both male and female. Common for both male and female. Like Turner syndrome is a common disorder found only in females. Klinefelter syndrome is a syndrome or an abnormality or a disorder found only in males. But this Down syndrome is common for both male and female. Our autosomes are common for both male and female. So this change in the chromosomal number, addition of one extra chromosome is to the autosome. So it is common for both male and female. What should be the chromosomal complement? Usually it should be 46. 46 is the balanced set of chromosome. I think in human beings all of us know 46 is the chromosomal number. Neither it should be 45 nor it can be 47. Here it is 47. Why it is 47? Because 2n plus 1. It is due to trisomy. So it was first discovered by a British physician by name Langdon Down in 1856. In 1856 by a British physician by name Langdon Down. Hence the name Down syndrome. Syndrome means what? Abnormality, anomaly. We use a word called syndromes, right? It occurs in 1 among 1,000 births. So it is a very rare. So only 1 among 1,000 births will have this syndrome called Down syndrome. And usually you find this Down syndrome usually in the babies born invariably to old mothers. What do you mean by old mothers? It's not a old woman, but the women who get late marriages. So who will have children after late marriages, the babies born to such women will have this Down syndrome, right? Now let us see what will be the chromosomal complement then here. So 44XX is equal to 46. This is the normal chromosomal complement. As I said, this is due to what? Addition, more trisomy, addition plus 1, and that plus 1 or minus 1 may take place to an autosome or to an allosome. Right. So here it is due to 21st trisomy. 21st trisomy means that plus 1 is added to the 21st chromosome. How many pairs of chromosomes are there? 23 pairs. In that 22 pairs are autosomes. 22 pairs are autosomes. 22 pairs are autosomes. So 1, 2, 3, 4, 5, like that 21st. So that plus 1 happens to this 21st chromosome. Hence, the chromosome complement will be 44, 45 plus XX is equal to 47. This is in females. In male, it will be 45 plus XY is equal to 47 because 44 plus 1 will take place. So, it will be 45 plus XX. So this is the chromosomal complement. Usually they'll ask, sir, if you want to write the competitive exam, this is asked. Give an example. So what is the example for autosomal hyperaneuploidy? So Down syndrome is an example for autosomal hyperaneuploidy. Autosomal means what? For the autosomes. Hyper means what? Plus 1 is added to the autosome. This is an autosome. So this is autosomes, right? So that is the reason why you call it as autosomal hyperaneuploidy. Hope you have understood about Down syndrome. Down syndrome is due to autosomal hyperaneuploidy. So what will be the chromosomal number? 44 plus 1 plus XX is equal to 47. 44 plus 1 plus XY is equal to 47. So this is autosomes. This is allosomes. This is autosome and this is allosome. So it is autosomal hyperaneuploidy plus 1. Right, now let us see. As I said, it was first time discovered by a British physician by name Langdon Down in 1856. Hence the name Down syndrome. Now let us see what will be the features of the affected individuals. How do we identify them that they are having this Down syndrome? There will be usually short stature, short. They will not be tall. Furrowed tongue. So the tongue will have one furrow, like the split. Then partially open mouth. So usually the mouth is closed, but here what will happen? Partially open mouth. And palm is broad with characteristic crease. Right? And physically and mentally retarded. They are physically, hence they are also called as what? Mongoloid idiots. Mongoloid idiots. Idiot means what? Whoever shows abnormal behavior is called as an idiot. So why they show abnormal behavior? Because they are mentally retarded. So they are also called as Mongolism. Down syndrome is also called as Mongolism. They look like Mongolism, a race called Mongolism. Right? So head may be smaller than normal. It is called microcephaly. Sometimes in some cases the head may be big when you call it as megacephaly. Cephaly means what? Head. So head may be smaller than normal, so hence you call it as microcephaly. What are the very important, this is a very common question which is asked in the exam. What is Down syndrome? What are the characteristic features of Down syndrome? Short stature, furrowed tongue, partially open mouth, palm is broad with characteristic crease, physically and mentally retarded, physically also, that is why they are having their short stature. Head may be smaller than the normal, hence we use the word called microcephaly. Then flattened nose, that is why you call them as Mongolism. Flattened nose, slanting eyes, then epicanthal eye fold. So the eyelids, no, it will not close properly. So they'll have this epicanthal eye folds. Small malformed ears. Then hands are short and broad with short fingers. Hands are short and broad with short fingers. Then short broad neck. Infertility in males. So usually it causes infertility in males. So all these are the characteristic features of Down's syndrome. So what are the characteristic features of Down syndrome? Short stature, furrowed tongue, partially open mouth, palm is broad with characteristic crease, then physically and mentally retarded, head may be smaller than the normal. When we use the word called microcephaly, if it is big, we use the word called megacephaly. Mega means what? Big. Cephaly means what? Head. Flattened nose, slanting eyes, epicanthal eye fold, small malformed ears, hands are short and broad with short fingers, short broad neck, and infertility in males. Moving on to the next very important syndrome called Klinefelter syndrome. This is due to allosomal hyperaneuploidy. This is an example for allosomal hyperaneuploidy. Here that plus, this is also due to trisomy. What do you mean by trisomy? 2n plus 1. Here that plus 1 is to what? To an autosome. Down syndrome, that plus 1 was for the 21st autosome. Hence 47 chromosomes. Here also it is 47. But that plus 1 is for an allosome, sex chromosome. Hyper means what? More, plus 1. So it is an example for allosomal hyperaneuploidy, and it is due to trisomy. And actually it was first time discovered by Harry Klinefelter, just like the Down syndrome was first time discovered by a British physician by name Langdon Down, and this was discovered, or first time it was discovered by Harry Klinefelter, hence the name Klinefelter's syndrome. So now let us see what are the very important features of this Klinefelter. So this is also very important from examination point of view, which is commonly asked in exam. In your second PUC board paper, these are the things which is asked from this chapter also. So all these concepts are very, very important from examination point of view. You should not take it too lightly. As I said, all the concepts what you study in second PUC biology is very, very important, not to just pass your PUC, but also to crack your competitive exams like NEET and CET. So the concepts are very, very important. Each and everything what you learn in second PUC biology is important. So you should not neglect, right? So what is this Klinefelter syndrome? It is the genetic disorder caused due to the presence of an addition of X chromosome resulting into a karyotype of 47, that is XXY. It occurs only in males. As I said, Down syndrome occurs both in male and female. Here it occurs both in It occurs only in males. So Klinefelter syndrome was discovered by whom? Harry Klinefelter. Right. Now, the chromosomal complement of Klinefelter syndrome is here 44 plus XX is equal to 46. This XY. This is common in male. Male, this is the normal male. But in Klinefelter's male, 44 plus X Y, instead of that one more X is added, it will become 47. See here, instead of only XY, it is 1 XXY. So there is an addition of one extra chromosome to the plus 1. So plus X is added to this. So it is, what is it? 44 plus XXY is equal to 47. This is a chromosomal complement of Klinefelter syndrome. So it is not added to an autosome. So autosome is normal. It is common. 44 it is. There it was 45. Here it is 44. Because there it was 45 in Down syndrome, because plus 1 was added to the Autosome. Here it is +1 is added to what? Allosome. So hence it is allosomal hyperaneuploidy. So instead of being 46, here you can see 44 plus XXY, that is equal to 47. So this is the chromosomal complement. There what was the chromosomal complement? 45 plus XX is equal to 47. 45 plus XY is equal to 47. This was the chromosomal complement for Down syndrome. See the difference here, right? So the chromosomal complement of Klinefelter syndrome is 44A. 44A means what? Autosomes are common. And 47. The chromosomal complement of Klinefelter syndrome. So actually in female it is 46. It should be like this. See in male how it is 44A plus XXY. So this is where this occurs only in male but not in female. So this is a very important concept to be learned. So what is Klinefelter syndrome? It is a genetic disorder caused due to the presence of an additional copy of X chromosome resulting into a karyotype of 47, that is XXY. It occurs only in male and it was first time discovered by Harry Klinefelter, hence the name Klinefelter. So what is a chromosomal complement? 44 plus XXY, that is equal to 47. In female it is common, so there is no change. But the change occurs in male. So there is one addition of one extra X chromosome to the normal set of chromosome. Here also you can see 1, 2, 3, 4, 5, 6, 7, 8. So this is common. 22 is common. But here one more X is added. Here you can see to an allosome. 22, 44 is common plus XXY is equal to 47. So autosome, there is no change in the autosome, but the change is only in the allosome. In male, instead of one pair, it is what? 3 chromosomes are there, X, X and Y. That is the reason why it is 47, right? Now let us see the next very important called Turner's syndrome. This is an example for, and this was first time discovered by Henry Turner. Henry Klinefelter. Their Down syndrome, Langdon Down discovered that. And Harry Klinefelter discovered Klinefelter syndrome. Here, Henry Turner, hence the name Turner syndrome. And this is an example for allosomal Hypo Aneuploidy. Hope you remember. And this is due to monosomy. Monosomy means what? 2n minus 1. So instead of 46, it will be what? 45. So 2n is what? Minus 46 minus 1, that is 45. Here it will be 45. And it is an example for allosomal hypo. Hypo means what? Less, minus 1. Hyper means plus 1. Hypo means minus 1, right? So it is an example for allosomal hypoaneuploidy. Now let us see what is this disorder. So Turner's, it is a genetic disorder caused due to the absence of one X chromosome and the chromosomal complement is 45 with XO. The syndrome is seen only in females, right? Now let us see. So as I said, it was first time discovered by Henry Turner. So here, this occurs only in females. It is called allosomal hypoaneuploidy and it is due to monosomy, that is 2n minus 1. So in females it is 44 plus XX is equal to 46. This is in normal females. So in normal females it is 44 plus XX, that is equal to 46. What will happen in affected females? What do you mean by affected females? Who is having Turner syndrome? 44 plus X 0 is equal to 45. What do you mean by 0 here? One X chromosome is missing. So what is it? 2n minus 1. So 1 is minus here. So here it is minus X. Here it is minus X. So what will it be? X0, 45. Instead of 46, it will be 45. As I said, this is only in female, occurring in female. Klinefelter syndrome only in male. Down syndrome both in male and female. Turner syndrome only in female. You should remember all this. And also you should remember the chromosomal complement. Writing this chromosomal complement is very, very important. It might be asked for 1 mark question or 2 marks question. Write what is the chromosomal complement of Turner syndrome? What is the chromosomal complement of Klinefelter syndrome? What is the chromosomal complement of Down syndrome? All these writing this chromosomal complement is very, very important. So Turner syndrome, it is a genetic disorder caused due to the absence of the X chromosome, and the chromosomal complement is 45. As I said, it will be 45 instead of 46 with XO. So one X is missing. The syndrome is seen only in the females. So the Turner syndrome discovered by Harry Turner, hence the name Turner's syndrome. So here the gamete, here also the gamete with 23 chromosomes. Here the gamete is with 22 chromosomes. So here monosomy. So it is, here you can see minus, so 1 is missing here, 1 is missing. So what will it be? 44 is common plus X0, that is equal to 45. Autosomes are same. Change occurs in what? Allosomes. Instead of XX, it will be X0. So 1 X is missing, that will be 45. Hope you have understood all the concepts what I have explained in today's session. So what will be the, see here you can see the child. So what will be the features? So as I said, 44A X0. Symptoms of the Turner syndrome. Now let us see what will be the symptoms. Broad chest, short stature, short neck with a webbed appearance. Right. So here you can see the child. Right. Puffy, it is swollen, face is swollen. So short neck with a webbed feet, webbed appearance. Then hands and feet may be swollen or puffy at birth. Infertility due to reduced ovaries. So as I said, this Turner syndrome is common in whom? It occurs only in females. Obviously, there will be infertile females. Why? Because the ovaries is not developed. And as I said, what do you mean by infertility? Inability to bear children. So heart abnormalities. All these are the abnormalities or the symptoms that are found in Turner syndrome, affected individual. So what are the characteristic features of this Turner syndrome? Broad chest, short stature, then short neck with a webbed appearance, hands and feet may be swollen or puffy at birth, infertility due to reduced ovaries, heart abnormal. The individuals having Turner syndrome are called sterile females. What do you mean by sterile females? The individuals who are unable to bear a child. So all these are the important characteristic features of Turner's syndrome. Hope you have understood all the concepts what I have explained in this very, very important chapter called Principles of Inheritance and Variation. This chapter also is very, very important from examination point of view. All the concepts you have to learn properly. Then only you will be able to answer any type of question that is asked in the board exam. Hope you have followed all the sessions what I have taught, particularly about this very important chapter called Principles of Inheritance and Variation. So in the coming session, I will be coming back with a very new chapter, chapter 8 called Human Health and Disease. I feel that this is the right chapter at the present scenario where we are all facing this coronavirus pandemic. So health is so very important for us. Maintaining a good health at this point of time is so very important for every individual on this planet Earth. And as you know, everybody is affected. The whole world is affected by this coronavirus pandemic. So what is human health? What is health and what is disease? All that we'll be studying in this very important and very interesting chapter. As I said, all the chapters that are in your biology syllabus are quite interesting. Only thing is you need to take interest in studying. So interest cannot be created. It has to come within oneself, right? Only when you have interest and you love the subject, then definitely you will learn all the concepts what is taught in this particular chapter. So I'll be back in the coming session with a new chapter called Human Health and Diseases. So till then, goodbye and thank you.